Hello everyone, my name is Joana Augusto and I'm here to present part of my PhD project about exercise as a neuroprotective strategy to prevent cognitive decline associated with old age. Lack of physical activity poses one of the greatest health risks to modern society, especially in relation to brain health. However, from a human evolution's perspective, humans are born to run. We are not adapted for the prevalent sedentary behavior that impairs overall health. The fact that our brains co-evolved with the muscular adaptations that enabled regular endurance exercise leads to the hypothesis that the human brain depends on regular physical activity to function optimally over the entire lifespan. Implicit here is the concept that the brain can detect signals from contracting muscles that modify the biology of its cells, and that the absence of these regular signals has functional consequences. For our first hypothesis, we suggest that single episodes of physical activity result in production of circulating molecules that cross the blood-brain barrier and modulate metabolic activity of brain immune cells, impacting on cognitive function. The risks of not exercising can become more severe with age. For example, it can lead to structural changes in the brain that often manifest as deterioration in cognitive function. However, there is direct evidence that exercise not only promotes memory but is also neuroprotective, and that these effects may be mediated by the induction of growth factor expression and processes like neurogenesis. Recent studies have also demonstrated that exercise helps reducing neuroinflammation associated with age. Despite decades of research in this area, the nature of the link between exercise and brain health is poorly understood. Studies have pointed out at lactate as a potential missing piece of the puzzle in this communication. Lactate is essentially an energy substrate and signaling molecule induced by muscle contraction that can play key roles in central nervous system function. For instance, it has been shown that lactate is neuroprotective while also promoting neurogenesis, as blocking its transporters inhibit these effects. Besides, lactate levels increase in both blood and brain during exercise. Altogether, these findings suggest that lactate may indeed be the link between physical activity and plastic change in the brain. Therefore, we hypothesize that multiple lifetime episodes of exercise cause persistent anti-inflammatory activity that is cumulatively neuroprotective. So for this study, we will aim to assess the impact of exercise on glial metabolism and cognitive function, while also examining whether exercise can modulate cognitive function and inflammation in the aged brain. And finally, assess whether lifelong exercise is neuroprotective and see if this is lactate dependent. Therefore, we will investigate the role of lactate throughout the lifespan of a mouse. We will do two main short experiments in which mice will be assessed for only 1 week, and one long experiment in which mice will be assessed for 3 times per week for a duration of 14 months. We will have a cohort of mice that will exercise versus mice that won't, so they will be sedentary and act as our control. In parallel to this, we will have a cohort of mice that will not exercise, but instead will receive injections of exogenous lactate. The reason for this is to see whether we can artificially mimic the levels of lactate observed in the exercising group. We will also perform behavioral tests in order to assess memory and learning. And finally, we will analyze metabolic and endocrine changes in peripheral tissues that result from exercise, while also investigating the changes in the mice brain. This study will represent the first time that recent discoveries in the field of immunometabolism and glial bioenergetics that centrally involve lactate metabolism are applied to the field of lifelong exercise-induced neuroprotection and long-term sedentary behavior. If the changes observed are in fact lactate dependent, we might be looking at potential development of exercise mimicking drugs.